Okay. Um, Seems like a lot of people are busy today. Anyways, let's get started. Welcome back to the Basis High School UCivil Club. Today we'll be covering Campbell's chapters 32, 33, 34. We probably won't get through the whole thing. I'll just try to move fast and um, finish as much as I can. So let's start with 32, an overview of animal diversity. Um, Here is a definition. Animals are multicellular, heterotrophic, with tissues that develop from embryonic layers. Yada, yada, yada. Let's cover some basics, lay some groundwork for this chapter. So animals reproduce through fertilization. Yeah, a sperm fertilizes an egg that turns the egg into a zygote. The zygote undergoes a process called cleavage, in which the you undergo repeated cell divisions without increasing cytoplasm, which you and the end result you end up with a hollow ball of cells. Think of it as there's one layer of cells um, surrounding a liquid interior. So the inside is just filled with liquid. The outside is like a single layer of cells. These cells are tiny, by the way. The zygote then the cleavage turns the zygote into a blastula. The blastula then undergoes gastrulation, that which turns the blastula into a gastrula. The gastrulation is, if you look to the left, you're forming the what's it called? Your digestive tract. You 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 think of it as imagine you have some kind of balloon and you push one finger in, except in this case you push it all the way through so that it comes out. It forms a canal throughout the entire balloon. Maybe a balloon's not the best analogy, but the thing is, gastrulation turns the blastula into a gastrula, which is, yeah. Development some and once animals finish their like the embryonic development, some are not fully mature. They're not adults. They're not sexually mature. Some have a sexually immature stage called the larva. And if you think, I'm sure we're all familiar with butterflies. The caterpillar is the larva of butterfly, and then it undergoes metamorphosis. And then the caterpillar develops into the adult, which is the butterfly. Anyways, let's talk a little bit about the history of animals. The general consensus for the origin of multicellular animals is there are unicellular organisms called choanoflagellates, and these they adapted a protein on their cell membranes that allowed them to stick together, and as the years passed, they somehow developed in a way that kept their multicellularity. So you have a previously unicellular organism that attaches to others of the same species, eventually forming a multicellular organism. Animals developed from choanoflagellates, and the first animals that developed in this way, well, the only ones anyways, the first animals to develop are called sponges. We'll get into sponges on the later slide. Here, a timeline. So I would just say this is a lot of, there's not much, there's no intuition here. Just know that it goes Neoproterozoic, then to Paleozoic, then to Mesozoic, then to Cenozoic. I would say roughly when the time periods start and end. Okay, Derek. Honestly, if you asked me just out of nowhere when was the Mesozoic era, I would have no idea. This isn't that important. Just I could tell you the general range. I can't tell you exact years. That's probably what you need to know. What I'm sure you need to know is the Cambrian explosion. And this was a period of time where animal diversity skyrocketed, possibly caused by an explosion in oxygen or like evolutionary pressures. We're not sure. But we do know that during these 10 million years, the we see a bunch of new species fossilized in the record. I guess no, just definitionally what the Ediacaran biota are. They're soft-bodied multicellular eukaryotes. These are like the first animal fossils. It's just a definition. Animals can be characterized by body plans. So all animals have some kind of body symmetry or lack thereof. They also have either 2 or 3 germ layers. Let's start with symmetry. So there are 3 types of symmetry. You have bilateral symmetry, radial symmetry, and asymmetry. Radial symmetry allows an organism to respond to the environment from all sides. Think of it as if you have eyes. Okay, Derek. Think of it as if you have. Imagine you have if you're radially symmetric. Imagine you have eyes pointed like forward, 120 degrees this way and 120 degrees this way. You can perceive information from all the directions. Similarly, starfish have they're radially symmetric, or roughly anyways. They have a number of arms that extend in every direction, and these arms allow it to perceive the environment from every direction. So these organisms are generally sessile, allows them to confront environment from all sides. Um, the other symmetry that we're going to talk about is bilateral symmetry. So if you look at the lobster down here, if you split the lobster down the middle, along I forgot how this works, along the dorsal ventral axis, or not along, but that way. You can see that it's symmetric. The 2 sides are like at least the exoskeleton is the same on both sides, and like we are bilaterally symmetric. If you cut an imaginary plane down the center of your body, uh, you're roughly symmetric, at least externally. Oh, thank you, Mary. Marie? Is it Marie or Mary? I mean, bilateral organisms are generally more mobile. They don't respond sessile. So the radial organisms need what? Wait, what did sorry, I'm sorry. I forgot which 2 options I said. Marie or Mary? Mary? Mary. Well, I mean, it has an RIE. I don't know what to say. I mean, okay. So Mary. Okay, you could have just said Mary, but it's not Marie. Oh my God. I don't know my short and long A. Marie. Mari. Mari. Okay, Mari. Okay, there we go. There we go. Cool. All right, back to this. Bilateral organisms, they're more mobile. So you have, think of it as, there are 2 trends that you generally see with bilateral organisms. One is called centralization, where the nervous system gets clustered in one place. So, for example, the majority of our decision making, higher brain function is right here in our head, right? That centralization, the clustering of the nervous system in one place or your head. The other trend is cephalization. So this is when the nervous system of an animal gets clustered, the nervous system and like sensory organs get clustered on one end of the body. So there is some overlap with centralization, but cephalization just means the nervous system and its sensory organs get moved to like the head end. So our eyes, ears, nose, they're all like above the neck, right? They're all on the anterior side. Yeah, so bilateral organisms are generally more mobile. Yeah. Now let's talk about germ layers. Germ layers are they're apparent in the embryo, and they're apparent in the embryo. They develop into a variety of different tissues. Every embryo has germ layers, but what's different is the number of germ layers. So you can have an organism with 2 germ layers, and that would be a diploblast. You can have an organism with 3 germ layers, that would be a triploblast. I think we'll get into the what's the word, the effects of it later. Not effects. I'm paraphrasing. Okay. Yeah. Body cavities. Now let's talk about body cavities. Animals either have a coelom, a hemocoel, or a lack of a coelom or acoelomate. The coelom is, let's talk about coelom first. The coelom is a body cavity that's entirely within the mesoderm. So like we talked about before, mesoderm is one of the germ layers. And in this case, we're talking about the adult tissues that the mesoderm turned into. Excuse me. So the coelom is entirely within the mesoderm, and the fluid within the coelom, the body cavity, cushions the internal organs. Like we are coelomates. We have a coelom that separates our intestinal wall from our intestine. And this helps to cushion our internal organs. It has a role. It physically separates the two. When your intestines undergo peristalsis, and when they pump food along, you don't feel it in your abdominal wall because there is a cavity between them that cushions the movement. Yeah, this coelom can also act as a hydrostatic skeleton for some animals, and a hydrostatic skeleton is, think of it as a skeleton is something that you exert a force on to move. Let's go back to the water balloon. Imagine you have a water balloon. You squeeze down on one end. The water in it is going to travel to this way, right? Squeeze down, water's going to go this way, and then when you release, the end of the balloon gets dragged along. So by like squeezing the water within the balloon, you're like moving the entire balloon in one direction. It is kind of, yeah, that's basically how it is. I have trouble wrapping my head around that concept, but you just squish one side and it goes. The second type of body cavity is called a hemocoel. And if you look to the right, this hemocoel is between the mesoderm and the endoderm. Above the the coelom, the body cavity was entirely within the red, entirely within the mesoderm. In the middle, it's the inside is yellow. That's the endoderm. Interesting thing about the hemocoel is that it's filled not with just interstitial or just like fluid. It specifically contains hemolymph. And I think it's worth getting into what hemolymph is. Hemolymph, let me first describe what we have. Humans and other mammals, and not just mammals, but we have separate circulatory and interstitial fluids. What that means is our blood, oh, okay, hold on, Marie, Mar, I'm so sorry, Mari, hydrostatic skeleton and liquid, I also feel all over space. Yeah. Yeah. I haven't pronounced hemocoel. Anyways, where was I? We have separate circulatory and interstitial fluids. And that means that our blood remains within our blood vessels, and our interstitial fluid, which is the fluid that bathes our cells, those two remain separate. Your blood doesn't go and mix around through your somatic cells. Some animals, like insects, for example, they have what is known as hemolymph, and in their case, blood and interstitial fluid they mix, right? There's no differentiation between them. They they mix into one thing known as hemolymph. I guess mixing isn't a great analogy because they were never the same thing. They were never separate to begin with. But there's one type of fluid that functions in both of these roles. So the hemolymph will carry nutrients and wastes. It will also bathe the cells. Hemocoel, yeah. And acoelomates like this planarian here, I think it's a planarian. It has no body cavity, and these are usually compact, flat animals. Yeah, I can't pronounce anything. I haven't pronounced these two words. Yeah. Now let's talk about let's talk about this in you typed it. Now let's talk about these two types of I guess two other ways to classify animals. So an animal can either be a protostome or a deuterostome. And what exactly these two mean, I'll get into in a moment on the next slide, actually. But here, a protostome undergoes spiral cleavage. So what that means is that instead of you have the embryo, imagine an embryo, and you have the vertical, horizontal axes. The cuts of cell division will happen diagonally to the vertical axes. So that results in asymmetric, yeah, do not let Ivan read. That results in asymmetric cells, like you see here on the bottom. Yeah, and they don't match up perfectly either. These protostomes are generally determinate. They undergo determinate cleavage. And what that basically means is that a cell's destiny is determined very early in development. There's in embryo, all embryonic cells start off as stem cells. They're totipotent, which means they can begin they can turn into any cell within the embryonic and adult forms of the organism. However, at a certain point, each a cell undergoes a termination where it decides it decides what pathway it's going to differentiate down. So once it makes this decision, it is no longer totipotent and cannot develop. It's no longer a perfect stem cell, right? In this case, in the case of protostomes, the cell fate is determined very early. So these cells lose their totipotency very early on. Deuterostomes, on the other hand, they have radial cleavage, and the planes of division are perpendicular or parallel to the vertical axes. This results in more symmetrical. This results in more symmetrical things, more symmetrical cells. These cells also tend to retain totipotency for much longer. Yeah, RIDS. I'm gonna be completely honest. What's RIDS? Help. It's not in the slide. It's something extra. Anyways, now let's talk about what protostome and deuterostome mean. So proto means first, deutero means second, right? And stome means mouth. So radial indeterminate. Oh, bruh, it's just an acronym. Oh, I thought it was something. Never mind. Well, if you want, yeah, there's an acronym. Yeah. So what was I saying? Proto means first. Stome means mouth. So protostome means first mouth. And what that means is that the first hole that forms in the embryo is becomes the mouth of the adult organism. So in this case, when gastrulation starts, when you start poking up, when you start the invagination, right? What? No, Derek. When you start the invagination, that forms that forms the first hole. That's the blastophore, and that becomes a mouth in a protostome. Deuterostome, on the other hand, wait. Oh no, you're right. I got confused. Deuterostome, on the other hand, means second hole, second mouth, and that means that so the blastophore will become the anus, and then when the canal pokes out through the other end, the second hole that forms will become the mouth. Yeah, cool. Views of animal phylogeny. Molecular and morphological data help to determine animal ancestry. That's basically what that's saying. For this slide, I would know that sponges are the basal group. They were the first to diverge from the rest of the animals. Know what eumetazoans and bilateria are. I mean, bilateria, it's in the name. They're bilateral organisms. And eumetazoa, you should just know that it's all animals except sponges. Bilateria is eumetazoa minus jellies and yeah, more. Okay. Lophotrochozoa and ecdysozoa. So animals are very diverse. These are two of the what's it called? These are two of the animal phylum, two out of many. I think I don't remember exactly how many, but these are two of the animal phylum that we will get into. I'll introduce them now because these are probably the most diverse of them. Start with Lophotrochozoa. Its name, Lophotrochozoa, is derived from its characteristics. So a lot of its members share either a lophophore, which is a crown of ciliated tentacles that function in feeding, or trochophore larva. You just know that trochophore is a type of developmental stage. Lophophore, you should know that it's a crown of ciliated tentacles. Lophotrochozoa. Yeah. Ecdysozoa. Ecdysis means the shedding of an exoskeleton, right? So ecdysozoa also derives its name from its characteristics. Ecdysozoans have an exoskeleton. So 33, introduction to invertebrates. This is all, there's no intuition in this. This is memorization. I guess I'll try to speed through these and get to the next chapter because this is just something you have to read a lot. Sponges, I should take it back. Sponges are kind of cool. There's a lot of detail in the sponges, but the others are kind of meh. So anatomy of a sponge. Here's the macroscopic view. A sponge is how do I describe this? Think of it as like a large hard sack, and the sack will draw water into its central cavity called the spongiocele, right? Sponges are filter feeders. As you see here, it draws water into the middle, like the cavity, and then it filters that water for nutrients. Then it pushes the water out of the top. And you might be thinking, how do they draw in water? How do they push it out? Is it like a muscle? Do they contract? It's like your heart? Not exactly. There are these cells called collar cells, and they have two defining characteristics. One is this long flagella, and the other is this collar with mucus that traps food particles. This is how the sponge filters its water for food, and hence the name collar cell. These are the cells that are very similar to choanoflagellates. As you might recall earlier in the slide, choanoflagellates are unicellular organisms that are believed to have like stuck together, and then when enough of them stuck together, they just form the sponge. Yeah, now let's talk about this cellular anatomy in more detail. Sponges have two layers of cells. They have what's it called? They have the inside, how do I approach this? One layer of cells lines the outside, and that's just epidermal. There's nothing really that too special about that. On the inside, lining the spongiocele are these collar cells. Like we just discussed, these filter, push water, yada yada yada. In the middle, there's like kind of, it's not really a layer of cells. It's more like free floating cells that can move around. Excuse me. These are called amoebocytes, and these amoebocytes build the mesohyl, which is it's a gelatinous layer. So think of it as like a cushion between the two cell layers. So it builds a cushion between the inner and the outer cell layers, and it deposits these things called spicules, and they're like skeletal fibers. I think they can be made of glass, which is why you should be careful around sponges. Silicon something dioxide. And these spicules provide some like support to the like overall sponge, because if you've seen some of the larger sponges, there are people who stick their entire hand into it. And if the wall was only 2 cells thick, then any kind of current could blow it away. Therefore, they have this mesohyl and these spicules that help to give it more support. Sponges are hermaphrodites. So sponges, they don't have a set gender. They start when a lot of organisms do this, where when an animal is smaller, it produces male gametes. It functions as a male. It like ejects sperm into the water. As the animal grows larger, it transforms into a female. And this is actually really interesting. The reason is because females, the the limiting factor in fertilization is not the number of sperm, it's the number of eggs. And the larger a organism is, the more gametes they can produce. So it makes sense for the smaller organisms to produce sperm, even though they can't produce as many sperm as a large organism, because no matter what, there's going to be more sperm than egg. When the sponge grows bigger, it can produce, it can produce more eggs. Yeah, I think that makes sense. Let me know if that doesn't. Yeah, they first act as one sex and the other. They diffuse both kinds of gametes into the water. Sponges are non-motile. I hope I don't have to clarify that. They find somewhere and anchor down, but their zygote, their larva, is actually flagellated. It can move around, find a new place to settle, and then once it matures, it, like its parents, becomes sedentary. So cnidarians. These are the jellyfish. They generally, it's just a two-layered sac. It has the endo and the ectoderm. It's a sac with a gastrovascular cavity. It has tentacles that can grab prey. It has two forms. One is a polyp and one is a medusa. So polyps are generally anchored to something, either to like a yes, that makes sense. Yeah, that's basically it. Yeah. Okay. Polyps either stick to a colony of polyps or they stick to some kind of substrate. The medusas are free floating. Doesn't matter which form it is, its anatomy is similar. It has a mouth, like one gastrovascular opening, tentacles that can grab prey, and special stinging cells called cnidocytes. These cnidocytes have nematocysts. You should just know, like, cnidocytes is the organelle that contains like the harpoon. The nematocyst is like the specialized nidai. Sorry, cnidocytes. I got confused. Cnidocytes are the cells that produce large numbers of nidae. Nidae is the organelle that contains nematocysts, and nematocysts contain a stinging thread that makes sting go burr. Movement, both inner and outer layers have contractile fibers. These cnidarians have a basic nervous system. They have a nerve net. They can help coordinate things. Yeah, and once they close their mouth, their medusas are basically like a water balloon. So they can use the fluid in the middle as a hydrostatic skeleton. Medusazoans and anthozoans. So a medusazoa, these are both still cnidarians. Medusazoa are just any cnidarians that can form a medusa. Scyphozoans, cubozoans, hydrozoans. Scyphozoans and cubozoans spend most of their time as medusas. Hydrozoans alternate between the two. You should know that cubozoans, also known as box jellies or cube jellies, they're very toxic, painful. They can kill you. Anthozoans, on the other hand, are cnidarians that only have a polyp stage. These are animals like coral and sea anemone. Yeah. Here is the life cycle of a hydrozoan, which is the alternating medusazoan. As you can see here, you start off with a colony of polyps, which some of them develop into a medusa. The medusas produce gametes. Gametes develop into more polyps. So it's like a circle thing. cycle. Okay, Lofa trochozoans played a molecular data widest range of body forms. Flatworms, flattened dorsal ventrally. I guess one thing you should know is that they don't have a gastrum. They don't have like a circulatory system. Their bodies are so small, so flat that they can gather nutrients and expel waste just by diffusion. They have a simple excretory system. They have what is called protonephridia. And these are also called flame bulbs because it's like a think of a light bulb with a cilia within it, right? And then the beating of the cilia draws water from the outside into like a duct. So you have like a bulb, a cilia, it beats and draws water into like a channeling duct. That cilia beating, people say it looks like a flame bulb. I haven't seen it, but I'll take their word for it. That the cilia is why it's known as a flame bulb. And this is like relatively simple. It's mainly used for osmotic balance. Cantinellida, chain worms. These are like worms that are attached to each other. Like the parent is attached to the child. Yeah, Rhabditophora will be discussed in 1 second. Most Rhabditophora are parasitic, but some are free living. It's not important. Okay, now let's talk about the parasites. Most of Rhabditophora are parasites. They generally have complex life cycles. They have alternating sexual and asexual stages. They generally need multiple hosts to complete development. Yeah, so like this schistosomiasis causing fluke trematode. Yeah, and it has to go through snails and humans, back to snails, yada yada yada. And now let's talk about tapeworms, everyone's favorite little worm. Very cute. These tapeworms of course, are parasites. They absorb nutrients from the host. They have a very long, tapered, and thin shape to maximize the surface area to volume ratio. It's important for them because they don't have any kind of digestive tract. They can't hunt for food. They get all their nutrients from diffusion. They have on their anterior end, so at their head end, they have what's called a scolex. And this is just a structure with hooks and suckers that help it like attach to the intestinal wall, right? Everything after that is just reproductive organs. So you have attachment, then reproductive organs. They do have, I mean, of course they do perform metabolic function, but their main job is just sex organs. And then they release the fertilized eggs into the feces of the host. Rotifers. So these are very paradoxical creatures. They're very tiny, so like very small organisms. They're smaller than most protists, which are unicellular. But unlike protists, they have complex organ systems. They're multicellular with, as you can see here, they have a digestive tract, they have a stomach, they have an anus. Oh, that's not that impressive. But they have a mouth, stomach, and then yeah, they have a hemocoel. They're like big organisms, complex organisms that just got shrunk down. I think maybe you should know that rotifers generally reproduce by parthenogenesis. Parthenogenesis is a process where usually the sperm of the sperm activates an egg. Usually when sperm fertilizes an egg and the nucleus enters the egg cytoplasm, the egg begins development into a zygote, right? But in the case of parthenogenesis, there is no sperm stimulus. eggs will automatically develop into a zygote. And this means that the females produce only females from unfertilized eggs. But during times of stress, like when there's a lot of rotifers in a certain area, population density skyrockets. That means that food resources are also scarce. Therefore, rotifers will sexually reproduce because those zygotes can remain dormant. And then when conditions arise, the new population, when conditions get better, the zygotes develop and the population goes back to parthenogenesis. Yeah, acanthocephalans. These are just specialized rotifers. They're also parasites, complex life cycles. Some creepily can manipulate their hosts. Parasites make crabs run in the open, so they will be eaten by birds. They have, I guess you should, like tapeworms, they also have a structure at their anterior end that allows them to attach better. Hook over. Ectoprots and brachiopods. So ectoprots are these two probably aren't that important. They both have a they both possess the coelom. Ectoprots, they resemble moths. They have a hard exoskeleton with pores through which remember lophophores are like ciliated crowns that help in feeding. They form like this peach yellow. I don't know what color that is actually. I don't know how to describe that. But the structure is hardened. It's like coral, and then it's extending lophophores like cilia crowns out of holes to feed. These are important reef builders. The other brachiopods, they're like clams, except their shells open up on a different axis, right? They're fake clams. Yeah. Okay, mollusks. These are kind of important. They're all soft. All mollusks are soft bodies. So like you might think, hey, snails have a hard outer shell, but their actual body is still soft. They just have they secrete a hard shell made of calcium carbonate. So here is their anatomy. They have a hemocoel, they have a reduced coelom. You should know that they have what is known as a muscular foot. This just allows them to move like our feet do. The visceral mass is just it's just what contains the internal organs. The mantle drapes over the visceral mass and secretes a shell. Mantle cavity is a water-filled chamber. So if you look at the diagram to the back, this mantle cavity is generally fluid-filled. It houses the excretory stuff, and the gill. A lot of mollusks have what is known as a radula, and this is like teeth-like structure, like a razor blade, that helps it obtain food. Mollusks are generally have separate sexes, but some are hermaphrodites. Chitons and gastropods. Okay, so these are classes of mollusks. Chitons have these dinosaur-looking things. They have 8 plates, but their actual body is one continuous thing. It's not segmented like an ant. Its muscular foot acts as a suction cup, and it grazes on algae. You should just know like roughly what it looks like. Chiton, it sounds old. Gastropods, these are like snails. They move either by cilia or rippling their foot. So they secrete a slime And they brush cilia over top of it to slide along. Some use their radicals to graze, others use them to bore holes in prey. So these radicals, they're sharp. They're not only used for herbivores. Predators can also use them. Bivalves and cephalopods. Bivalves are clams. They have a shell that divides the organism into two halves. The shell is open and closed by an adductor muscle, and this muscle is very powerful. It's a filter feeder, and some bivalves, it looks like a very demented butterfly. It can open and close its shell in a way that looks like it's flying. You can search it up. Actually, quite funny. Yeah. Cephalopods. These are squid, octopi, very active, fast predators. They grab their prey with tentacles, and they have poisonous saliva. It's cool. Their foot has become the excurrent siphon, which think of it as like jet propulsion. Um, you just you like drink. Imagine if you could like drink water and like shoot it out of your hands, like Iron Man to move somewhere. That's basically what they do. Except it's not a hand; it's a foot. Kind of gross. Most cephalopods have lost their shells. Um, they have a closed circulatory system. Blue blood. That's cool. Uh, they lost their shell. Like octopi. Have you seen an octopi with the shell? They generally need they're predators. They need to be fast, and they're not as afraid. They're not that defensive. And yeah. Mollusks are going extinct. Um, I think Campbell's makes a note that the sixth mate we're going through the sixth major extinction event, and mollusks make up 40% of all extinct species. They are at high risk for going away. Very sad. Yeah. Anyways, annelids. Let's talk about these guys. They're segmented worms. They have a coelom, and many have just know they have like bristles made of chitin. Um, Arantia sedentaria, marine worms, Pteropodia. You can just read about this. There's not much that I can say. Excuse me, other than what's written here in the book. Leeches. Leeches are they're called sedentarians. They're sedentary. Um, they're they're a lot of them. We generally think of leeches as blood sucking. That's I guarantee this is what popped into your mind when I said leeches. Many are actually predatory. They hunt their prey. Yeah, parasitic leeches. They secrete compounds in their saliva that prevent the host from knowing, one, that it's been bitten, and two, that it stomach cephalop. Mm-hmm. Yeah, those are the derivatives of parasitic leeches. Leeches secrete an anesthetic and slits the skin of its host. It applies an anticoagulant to keep blood flowing. Um, if you get a cut, normally your clotting factors will prevent the bleed from continuing. Leeches secrete an anticoagulant that that stops the clotting. Hence, it can drink a lot of blood, and it can drink a lot of blood. It can drink almost up to 10 times its body weight. And leeches they're used clinically. Um, they can suck out hematomas. Hematomas are like a hema, blood, toma. I think it's like a body, or like I don't think blood tumor is the right word. but it's like a accumulation of blood, basically. Um, it's it's anticoagulant is also used clinically as a blood thinner. So like if you haven't, if you, and never mind, I don't remember how you get it. There's many ways to get it. Anyways, second class of sedentarians, earthworms, they they eat soil. Um, they're decomposers. The slime that they secrete helps to aerate the soil and improves the texture. They're hermaphrodites and they can reproduce by fragmentation. This is an Australian earthworm. Everything's bigger in Australia. And as you can see here, earthworms are actually very complex creatures. They have start from the top, mouth, and this is kind of like their brain area. Actually, excuse me, give me one second. Oh, sorry. So this ganglion is kind of like their brain. They have a nerve core stretching down. Um, Opens up in the pharynx, esophagus, crop. I think I always get the crop and gizzard mixed up, but the crop, I believe, is where you like store food, you moisturize it, and the gizzard is, or maybe the crop is also where mechanical pulverization happens. Uh, I'm not sure, but in one of these places, they sometimes carry like little grains of sand or rock that help grind up the food. Uh, I'm gonna move on. So, ichthyozoans are the most species-rich animal group. So, like I was talking about before, these are the ecdysis, the animals with exoskeletons, very diverse. First, nematodes. They have a tough cuticle. These are like roundworms, I think. I think that's what they're. I'm not. I don't. They have a tough cuticle. They have no circulatory system. There's hemolymph and their hemocoel. They they wiggle forward, in like a weird undulating fashion, kind of serpentine. As you see here, the muscles are oriented along the dorsal ventral axis. And so if you contract one, it like bends the body. It bends the body like one way or the other. Uh, all body muscles are longitudinal, moves by wiggling forward. So important you can. Uh, oh yeah, you should. If you're gonna take away anything from the slide, maybe know that probably just know about trichinella. You can get this parasite by eating undercooked meat. These parasites mature in the intestines. Oh, these are gross. Um, so you can get it by eating undercooked meat, and then they develop in your intestines, and they burrow into the intestinal wall. Um, then they migrate, and under a microscope it looks really gross. They can migrate through your body to different parts, and they generally insist in like in muscles, and you can see like little pockets where they are. It, I mean, if you want, you can search it up. It's kind of cool, also kind of gross. If you just ate, I would not suggest. But if you have an empty stomach, I would recommend. Um, oh yeah, another thing, they have an angiogenic effect. They can direct arteries to they can grow blood vessels towards them to bring more supply. How much more do I have? Yeah, like I said, there's just a lot of classification. I think I will. I'll introduce arthropods and then we can pick up with this next time, or I'll post the rest of the slide. Yeah, okay. So arthropods, these are they're very successful just in general. There's like this many arthropods. I don't even know how many that is. Oh, did I do some? Oh, um, this is, oh, by the way, arthropods are a class of ecdysozoans. They have a segmented body, they have a hard exoskeleton, and jointed appendages. Uh, I, I I'll just get in. I'll just introduce this idea now. So a lot of arthropods have like many different like segmented arms, and this is important because with so many arms, it's easy to repurpose mm, think of it as if you had a lot of like spare, if you have a lot of spare fingers, and then you can evolution will start playing with your spare fingers and like giving them new functions. Like maybe one finger becomes very sharp, or the other one like develops a toothbrush, or I don't know, but they can develop novel functions simply because there are so many of them. Segments can divide, labor, and specialize. Like for example, in a lobster, the claws developed from appendages similar to the legs, or they were once similar, and they diverged. Appendages come in pairs, tough exoskeleton, and these are some of the characteristics that made them really successful. They have a strong defensive system, tough exoskeleton, and paired appendages just give them a lot of utility. They can do a lot of things. Yeah. Okay, so I'm going to end off there.